The overall goal of this transmigration assay is to investigate lymphocyte extravasation into the central nervous system in vitro.
This method can help in furthering our understanding of inflammatory diseases of the central nervous system and in the development of new therapeutic approaches.
Some advantages of this technique are that it is easily accessible and that it allows analysis of rare lymphocyte populations on single-cell level.
This method can provide insight into lymphocyte transmigration across the blood-brain barrier and can be applied to the study of transmigration via the blood-cerebrospinal fluid barrier.
In addition, Evans blue permeation analysis can be performed to assess the integrity of the blood-brain barrier model.
Begin by covering the bottom of a T25 cell culture flask with 2 milliliters of freshly prepared fibronectin solution for an at least 3-hour incubation in a cell culture incubator at 37 degrees Celsius and 5% carbon dioxide. At the end of the incubation, replace the fibronectin solution with 7.2 × 10 to the 4th human brain microvascular endothelial cells, or HB-MEC, in 6 milliliters of complete ECMB medium, and return the flask to the incubator. When the HB-MEC reach an 80% confluency, transfer the supernatant to a 15 milliliter conical tube and wash the adherent cells 3 times with 5 milliliters of PBS. After the third wash, add 2 milliliters of pre-warmed Accutase solution to the cells for 2 minutes at 37 degrees Celsius. Then firmly tap the flask several times, confirming the cell detachment by light microscopy.